Little introduction to the top level of today's topic, which is about metagenomic analysis. So that means looking at every possible gene at the same time, and we're looking at every gene without necessarily knowing which organism the gene came from. We're just going to take all the DNA, wherever it happens to be, inside of a gram of soil, for example, and read out what all of it says at the same time. So we'll get a kind of a picture of the total genetic repertoire of this group. And it's looking at the microbial community, so we're looking at specifically bacteria, and I don't think they measure archaea in here, but we always ignore them, and yet they're actually really important as well. So microbial community and function involved in, this is cadmium metal, and I don't know why they're weirdly capitalizing it. Actually, that one's okay, that one's not okay. But cadmium contaminated soil is something you would find, for example, around factories that have manufactured nickel cadmium batteries for a long time. You tend to get some really strong soil contamination around those, and it's very, very poisonous. And so there are a lot of people that are trying to find some kind of microbial solution, some kind of combination of microbes that can go in and detoxify this soil. Either sort of pack all the cadmium away inside themselves and take it somewhere else, or convert the cadmium into a form where it doesn't actually cause any damage to other forms of life. And so you're going to have a look at this paper, and this is my goal, for you to look at this paper and read it and be able to understand it, and this is what I would like you to discuss if you get this as a test question. So these tables are going to show you the properties of the different soils. They're going to show you a summary of the metagenomic sequencing, so let's see what that looks like. There we go, that's just how many things they read. So the raw reads, this is how many sequences you can get in a single run that just costs a couple $1,000s. That is 100 million sequences. Fantastic. And it looks like they might have done this twice, and they got 100 million the first time and 94 million the second time. Oh well, yeah, slight drop off. And they're going to be small pieces. They're going to be around 500, 550 bases. And you'll have a couple of the bigger ones that'll be around 1,000 bases for that context. So that's pretty good. That's gives you a lot of information. Yeah, all at once. Okay, so let's go back to the article. Yes, please. And there's another table. That's right, there's two tables. And then here, this is going to be a useful one. So this is summarizing some of the genes that were found. And I'm going to let you look through this. But look at them. So different nucleotide transport, metabolite biosynthesis, something like that, cell motility. Yeah, these are a repertoire of genes, and a lot of these genes are going to be just normal things that keep bacteria alive. But if you find an unusual gene that seems to be common in cadmium-containing soil and is not found in other places, then the idea would be you try to express some of those genes, so maybe in this function unknown or general prediction only category, and you try to see if any of those might make an enzyme that would actually solve the problem for you. And that's going to be the idea of this. This is going to go through the different kinds of bacteria, and it looks like there are two different sites, site 1 and 2. And this is just at the phylum level, what kind of bacteria do you see? And yeah, there's a lot of different kinds in there. Similar ratios, but not identical among these different samples. And that is also always the case. And this is comparing the two sites, seeing which ones are more contaminated or more abundant or less abundant. And you see there's a couple of them that are much more abundant at one site than the other. And so from this, they're going to use this to actually build up a picture of some of the bacteria and some of the genes that might be very useful in cleaning up cadmium-contaminated soil. That's the idea. And again, I think this is an interesting topic. This is something you're going to want to talk about with other people that you know. And so now you've got a paper that you can do that with. So thank you very much. This is our little intro to the paper.